So hello, my name is Joshua Johnson. I'm a second year graduate student in this lab, and we are in the Hakamara lab, which is in the Lee Ka-shing building. It's where we do our science. So I'm from South Carolina, in a rather small city called Spartanburg. And my parents were sort of blue-collar family. So my dad was a supervisor and a manager at this injection plastic molding plant, and my mother was a bank teller, but she then became a stay-at-home mom as I became older. I always thought science was pretty cool throughout high school, but I was never a very driven student during high school. But when I went to undergrad, originally with the intention of going into medicine, I started taking science classes that were taught very well at very good professors at the university I went to. And I became just in love with science and the idea of doing science, critical thinking, and using that to understand nature. And so that's why I'm continuing my studies here at Berkeley to hopefully get a PhD in this subject and continue doing what I love. So our lab has two main aims, one of which is to understand the biology of telomeres and also of telomerase, which is the enzyme that maintains these telomeres, which are the ends of chromosomes. And also another aim of our lab is to understand how cancers can form from sort of normal cells, what sort of genetic events occur to allow that progression to happen. In order to do that, we study human cells and culture, and then we do different genetic manipulations in order to see what sort of effects we can get in order to parse out the biology behind these very important things. But our aims really are heavily related to understanding cancer and also aging. So I mentioned before that we do a lot of cell culture. So in order to do this, we have to keep this sterile to make sure, so we have a lot of nutrients on our cells. And if bacteria or something gets in there, they'll use those nutrients too, and that'll be quite bad for our experiments. So we have to do all of our work with our cells in what's called a tissue culture hood that allows us to have an environment that's sterile, that we can do our experiments with our, do manipulations with the cells without, fear of having them getting contaminated. So that's where a large amount of my work is going to be just inside the hood. But also a lot of the work happens also on the bench. So when we take our cells, we collect samples, maybe we collect DNA or we collect proteins or RNA or something, we could manipulate with them on the bench top to sort of figure out what's going on inside of them, what genes do they have, what sort of what sequences do they have. So we figured that out usually on the bench here by doing different experiments. So working in the lab really does engender sort of critical thinking with looking at sort of the different aspects of your science, but it's also neat when you're outside the lab to take that sort of same philosophy of critical thinking and to look out, out of the entire, just all aspects of the world. And so it does allow you to be just a bit more critical or more skeptical about things, and then it definitely affects the way you interact with the world, and I think in a positive way. So I just enjoyed the aspect of asking questions where the answers were not known, and then having to sort of design experiments and work around those things in order to understand something that we just haven't understood yet. And that was just a fascinating notion that I didn't see being the case in medicine. And so I just really enjoyed sort of the intellectual puzzle solving aspect of it, which is why I decided to go this route instead of the medical route. So this lab it's a work environment for sure, where I come in to do all the things necessary to move my experiments forward. But it's also quite a fun social environment too, in our lab especially. So we have a very diverse lab with people from multiple backgrounds, and we all get along quite well with each other, and we do have a great time working with each other, and we get to bounce ideas off of one another, because no matter how smart you are, if you're just thinking from your own perspective, it's going to be quite limited. So it's an amazing environment here to be able to communicate with other people that are doing projects that aren't exactly like yours but very similar to yours, and then you can get different ideas from things like that. It's a lot of fun to be in that sort of space. So my advice would be to be very active in your own education, and then to really seek out and reach out to the faculty whom you think will provide good mentoring, or senior students or graduate students, depending on where you are, and then just really being an active force to going to them, and then asking them, and not being afraid of being ignorant, because you are going to be ignorant in the beginning, and it's okay. And then, but the way to escape that is to be very inquisitive and be very active and trying to figure out what you're interested in. And then once you do find that, you can really go at it, and it's quite a lot of fun. You can learn a lot. So, yeah.